Hey, warm welcome to this video. Now, I've been looking forward to this because I need my science brushed up on a bit. We're talking to Dr. David Speicher, who's the virologist in Canada, who's done the analysis on the RNA vaccine samples from Australia. So, David, thank you for coming on and welcome.
John, it is such an honor to be here on your show.
Not at all. Thank you for coming. So you were sent these vials from
Yeah.
these vials of vaccine from Australia to analyse them for DNA contamination. But there's a background here, isn't there? DNA contamination has been found in RNA vaccines before. They're not supposed to contain DNA, but it's been found that they do, and you've also replicated that finding. Is that basically what's happened?
Yes, the first finding of this is for Kevin McKernan last year February, and basically Kevin is a genomics expert, does whole genome sequencing, and did a DNA negative control and said, well, I'll use a COVID vaccine as it should be, just modified mRNA and a lipid nanoparticle, and that's it. And his sequencing started to come back with all sorts of DNA. And it's like, this isn't supposed to be here. He looked at a small sample set and was criticized for it, and so I was asked to help confirm his findings. I've looked at so far 32 vials in
Well,
Canada. I've looked at all the original Wuhan, the monovalent, the bivalent, and the XBB 1.5 boosters, and have found DNA in all of those vials, and all of the Pfizer vials contain an SV40 enhancer and promoter. So based on that work, we shoved a print out last October on the first 27 vials. A full paper is about to be submitted, and so based on that work, I was asked to test vials from Australia.
And you found the same DNA contamination in the Australian vials as you had in the other
Yes,
vials from other countries.
except a much higher level.
Now, these vials had to be transported from Australia. Is it possible that this DNA could have somehow materialized when it was being transported and it wouldn't have been there in the clinical situation, or has the DNA got to have been there all the time?
No, it was there the whole time. It's technically not DNA contamination. It's a residual plasmid DNA. It's a process impurity, and it's because the DNA plasmid in process 2 was to take a DNA plasmid, grow up that in E. coli bacteria and make millions of it, extract just the plasmid part out of those cells, and then use it as the backbone to make all of the modified RNA.
Ah, I see. Right. So it's
Okay.
the E. coli plasmids being used as almost the recipe for the
Yes.
mRNA.
Yes, it's a synthetic plasmid picked from the Pfizer's division of gene therapy, and it's a gene therapy plasmid used to make the modified mRNA.
That makes sense because the central dogma of genetics is DNA makes RNA makes protein. So
Right.
it kind of follows on. Does it matter that there's DNA post-processing impurity in the vaccine?
Well, it should have been removed. They have tried to break up this with a DNAs, an
enzyme to break it up.
which breaks up, and therefore all of these are all fragments, and so normally if it's free DNA, it wouldn't be as big of a deal, except these are all packaged inside the lipid nanoparticle, and therefore are injected or transfected into the cells in our body. Are those ovary cells, liver cells, heart cells? The whole biodistribution work of Dr. Byron Friedel showed that the shots do not remain at the site of injection. So
So the lipid nanoparticles ensure that this RNA is transported around the whole body, your liver, your heart, your ears, your toes, everywhere. But packaged in those lipid nanoparticles is the DNA post-processing impurity. That pretty well guarantees that with the RNA, the DNA impurity, these plasmid fractions, is going to get into potentially billions of individual body cells.
Yes.
And what happens to the DNA once it's in the cell? Because if it goes into the cell, that'll be in the cytosol of the cell, the outer part of the cell, won't it? Does it do any damage there, or does it migrate into the nucleus, into the inner part of the cell where the body stores its own DNA?
Well, that's a very good hot topic, especially as these fragments contain a SV40 and enhancer.
And what we're concerned about the SV40 enhancer is that means it's much more likely that this contamination will get from the cytoplasm into the nucleus of the cell and promotes the likelihood of it integrating into the body's own DNA.
If I may share my screen,
please.
If we look at the 1999 work of David Dean out of University of Rochester, if this is the data box which transcribes is the start point of transcribing DNA into RNA. If you look at this abstract of the paper, and this is on DNA fragments, and the key parts are in yellow. If you take DNA fragments with a 72 base pair repeat of the SV40 enhancer, it'll facilitate maximal transport, and this occurs before at times before cell division, as few as 3 to 10 copies per cell of cytoplasmically injected plasmids containing the SV40 enhancer, like a lipid nanoparticle injects the DNA and RNA pieces into our cell. If you have as few as 3 to 10 copies per cell, it will greatly increase their ability to be transported into the cell's nucleus, especially in non-dividing cells, which means it'll move those DNA fragments across the nuclear envelope, and it could integrate into the cell. Is integration possible? Well, Kevin's work in the past months has shown one case. If you take cells in cell culture over the vaccine overnight and wash off the vaccine the next day, and then harvest those cells and sequence the cells, it's been found full length spike DNA pieces in chromosomes 9 and 12. So
Wow.
in cell culture, it's been seen. Kevin's Substack this week showed in a cancerous tumor a year post vax, the spike DNA is still present in a cell.
You know, this really answers a lot of questions, because I had assumed that the spike protein getting into the human cell nuclear DNA must have been the result of reverse transcriptase.
That's
not
also possible, and so is this as well.
But so what you're saying is it's not the spike protein RNA, it's the genetic information which originally coded for the spike protein RNA.
Right.
In the manufacturing process. That genetic instruction to make the spike protein in DNA format gets into the cell, that gets into the nucleus, and that can integrate into human nuclear DNA. And that has actually been demonstrated in human cell cultures. And it's been identified in chromosomes 9 and 12. That means, in clear terms, it's induced mutations in chromosomes 9 and 12.
Yes.
And cancer is a mutation.
Mm-hmm.
Wow. So that's actual scientific evidence that this occurs.
Yep. And I'll show this, and this is from Kevin's Substack.
Yep. Yep.
And he has sequenced full length spike integration in chromosomes 9 and 12, and one of these is in a centromeric repeat, and the other is in a pre-cancer gene.
Right. So this one is specific to a potential what one is a mutation in an actual oncogene.
Yeah.
And what's the centromeric DNA? Is that the little bit in the middle?
Yes.
Is that particularly significant from a functional point of view?
It might be. That's beyond my area of expertise.
Right. But we know, but they know there's mutation in a potential oncogene.
Oh yes.
Well, now, how do the regulatory authorities respond to this? Do they have they accepted this science, or are they saying, well, what are they saying? The science seems pretty terrifying to me, to be quite honest.
Well, Health Canada has said very little. Last year in August 10th, the Epoch Times emailed Health Canada, and at first they said that any claims that the presence of a SVB 40 sequence is linked to cancer is unfounded, and that they at first said that the Pfizer vaccines don't contain sequences of the SV40. They went back and looked and found it, and in the House of Commons, on a order paper question, MP Colin Kerry asked about the SV40, and Health Canada said on record the fragment is inactive and has no functional role, and was measured to be consistently below the limit required by Health Canada. At the same time, in a ATIP by Goops Migu of to Health Canada, we see a email sent to Pfizer on August 4th. Health Canada says, why is this SV40 sequence here? What's the justification of it? And what is the size distribution of the residual DNA fragments? And so Health Canada, FDA, TGA, EMA all know it's there, and so far have been pretty silent on this whole thing. It's like, it's there, it's no big deal, whatever. And I've shown in the David Dean paper that this is a big deal.
But all the information here is redacted. So this well, what's an ATIP, David?
What's that?
What is an ATIP?
Is a access to information
Right.
Asked.
It's like a freedom of information request.
Yes.
Right? So so Health Canada have sent this out.
Mm-hmm.
But it's so, well, why have they redacted all this information? What have they got to hide?
That's well, what I would like to know too. Rebecca Barnett in Australia asked the same thing of the Australian TGA, and received 72 redacted pages. Every page was redacted.
So it
And
sounds like Health Canada and the TGA in Australia are both hiding the same information.
exactly. Yeah.
But I want to know.
And
This is outrageous. I want to see what's behind these black bits.
so would I.
Absolutely incredible.
Right?
Yep. Now, the technique that you used, David, it was described.
Yes.
The technique was. I'm not familiar with it at all. It's fluorometry, is it? What does this technique involve?
Thermometry
Philometry, yeah.
is is a inch you use as a instrument, and so in a tube you mix DNA dye like you're a green ribogreen in the tube. You add the DNA or your sample into the tube. The dye binds to
Right.
DNA, and therefore, if it binds, if it's excised with a the light, it'll express and it'll fluoresce.
Right.
And based on that, you can tell how much is there.
So
So
basically, you're making the DNA fluorescent.
yes, yeah, and it works on total DNA. And so if you use PCR, it's a amplification of a specific fragment of the DNA. This fluoresces all of the DNA in that sample. Now, when I looked at the Australian vials, okay, I looked at both PCR and thermometry, and so this is quant PCR. This is qubit here. On the left at the bottom is the PCR results, and if we compare this to the TGA, EMA, FDA cutoff of 10 nanograms per dose, and this is a guideline for naked DNA in a normal vaccine and not in a lipid nanoparticle, and it's supposed to be less than 200 base pairs in length. Average size we have found is 214 base pairs. The largest fragment is 3,500 base pairs. And so if we look at Pfizer, the purple top, and see all of these appeared and came to me as a sealed vial, except the Moderna. And so these are all intact, sealed vaccine vials. It showed up. The dry ice was gone, except DNA is very stable at room temp for months.
Oh yeah. Well, we
And-
dig up
and-
DNA from bodies from hundreds of years ago, don't
Exactly.
we?
Exactly. And therefore, if it did anything, the levels would have gone down and not up.
And
And
the
so-
DNA can't spontaneously appear. It's got to
No.
have been there at the beginning.
Exactly. And so Spike and or in the SV40 are all above 10 here. This vial, the Spike is above, and in Moderna it's just under. Except if we look at fluorometry of total DNA, we see it's like 1,000 to 500. And so what I did to explain things in a simple term, if you take the vaccine as is in the vial, you have in there RNA and DNA. If you're using a dye and you've got excess RNA in that sample, like in these vaccines, there is crosstalk, and the dye could stain up both. And so if we look at that, we get the red bar. If we boil it, we basically lyse all of the lipid nanoparticles, open them up, expose what is inside it to die. That's how come this spikes up. And then I add RNAs to basically wipe out all of the RNA in the system and get only DNA. And so all of these grains are just total DNA found in that vial. In the Pfizer's, the total is about 800. And keep in mind, these are nanograms per dose. So this on the left is a adult dose. This is and in the middle is a child's dose, which is a smaller vaccine amount, less DNA. except it's still above 10. And Moderna is well over 1,000 nanograms per dose.
So the basically, I think you're saying that the fluorometry is a more accurate technique for quantification of total DNA present.
Total DNA.
Yeah, and you're clearly showing that, and you're confident that these results are correct.
Oh,
But
yeah.
also, I think the thing that came through to me there, David, was that these guidelines for the amount of DNA that's allowed don't allow for the mega superbly efficient delivery system that the lipid nanoparticle is.
Exactly.
So small amounts of DNA in a muscle cell would just be broken down or phagocytosed. But if that's
Right.
systemically distributed, so really the levels that are set for lipid nanoparticle vaccines need to be essentially zero. They need to be 100 times less than the limit that's currently set, because the limit that's currently set doesn't allow for lipid nanoparticle vaccines. That makes it even more alarming. And given that you only need, I think 3 to 10 copies
Yes.
per if you've got 3 to 10 copies per cell, that can get into the nucleus, and we've got these massive amounts. It sounds to me like transport into the nucleus is essentially inevitable because of the combination of the efficient lipid nanoparticle delivery system and the SV40 causing it to go into the nucleus of the cell from the cytosol of the cell.
And two, if you look at the PCR results, okay, those on Pfizer in red are a CT between 10 and 12. Okay, which is about 10 trillion DNA copies per dose.
Wow. So 10 trillion per,
With a T.
a trillion being 1,000 billion.
Yes. Which is
So 10 trillion copies per dose. We only need 3 to 10 copies per cell.
yeah.
So you've got the possibility there of infecting at least 1 trillion cells potentially.
Yeah.
Wow. Yep. Okay. That's interesting scientific information. So once the DNA is in the nucleus of a cell,
Yeah.
how does it get into the DNA? Is there an enzymic facilitation, or does it simply diffuse into the existing DNA sequences?
That's a very good question, which I don't know, John.
But we do know it happens because
Yes.
Kevin's shown that it goes into chromosomes 9 and 12.
Yes.
So we know that it does integrate into host nuclear DNA, causing mutation. Now, if this was in a germ cell,
Yep.
could that potentially alter the genetic nature of the next generation?
I will find out in the next 6 months, hopefully.
But theoretically, that possibility is there, isn't it?
Yeah,
Because
it's quite theoretical. And two, if it did that and made a ring, every cell in that child's body could potentially make spike.
And that could be potentially passed on through, because if it's in the DNA of the zygote of that
Yes.
potential child, then it would be in the germ cells of that child and be passed on indefinitely.
Yes.
Wow.
Which I hope we don't find, because that is a very alarming discovery if it is found.
But it is a theoretical risk.
It's a theoretical risk.
So potentially spike protein could be getting produced by all future descendants of that individual.
Mm-hmm.
Wow.
And two, it also means that spike detoxing is also very difficult if your cells keep making spike.
People with long COVID type syndromes, and we know that people with post-vaccine injury have long COVID type symptoms.
Yep.
That they seem to have ongoing production of spike protein. Does it sound reasonably possible to you that this ongoing production of spike protein could be caused by the SV40 DNA integrating into the nuclear DNA of the cell? The nuclear DNA of the cell, which is now mutated with the SV40 and the spike protein production sequences, that they could then transcribe that into the body's own RNA, that could then go into the cytoplasm to be translated into making the body's own ribosomes to produce spike protein that's then excreted from the cell, resulting in the ongoing production of spike protein.
It's very possible, John.
Because it seems very likely that people with some forms of post-vaccine injury are still producing high amounts of spike protein.
Yes.
And that is unlikely, in my view, to be caused by the fact that the RNA would hang around in the cytoplasm for a year or so. Much more likely that it's coming from fresh transcription from the DNA. I mean, is that your understanding? Is it unlikely that RNA could hang around in the cytosol of the cell for weeks, months, or a year?
It is possible for months. I don't think it's possible for, long term.
Yeah, even months surprises me. Interesting to hear you say that. I would have guessed days myself, but if you think it could be months, then.
See, the downfall with these is that it's not just mRNA. It's modified RNA.
Yes. Of course.
With a N1 methyl pseudouridine, which makes it much more plastic and much more resilient.
Yeah.
And two, because of that, it's prone to ribosomal frame shifting.
Right. So the fact that the RNA is modified means that the way that the code is translated into a protein could be shifted a base or two along, and that's going to change the sequence of amino acids.
Exactly.
And that means the body could be, if you like, conned into making a completely novel protein.
Well, As well, it's making short length spike, mid length spike, full length, and so it's a wide range of aberrant proteins being made, and therefore it'll throw off our whole immune system as it's looking at short spike and long spike and middle spike. And
so the ongoing problems that people with vaccine injury have could be the body's immunological inflammatory reaction to short bits of spike, medium bits of spike, longer bits of spike, basically a polyclonal spike.
It's possible.
Causing inflammation throughout the body.
Yep.
That's possible.
It's possible.
Wow. Amazing.
So that's
yep.
And that doesn't even look at the whole possibility as well in these vaccines as double-stranded RNA, as well as possible RNA DNA hybrids as well.
I didn't know you could get double-stranded RNA. Right. Okay.
Yes.
Right. Wow. And what the heck does that do? Do we know?
That's a good question, John.
Yeah. And we could get like a strand of DNA and a strand of RNA somehow combined as well. Is that?
Yeah.
I've never heard of it. Right. Okay. Fine.
Yeah.
And again, what the heck does that do?
Yeah.
Wow. So, given all these unknowns, should we still be injecting people with RNA vaccines?
I don't think so.
Neither do I.
Because, are they safe? Are they effective? Safety has not been shown, and they're effective at causing harm.
Mm-hmm. Yeah. So when I first heard of these vaccines, it was just inconceivable to me that a vaccine could go on causing damage for months and years ahead, and that has just completely changed with these bizarre genetic preparations. And I think I was conned by the fact that people called them vaccines, but they're not. They're genetic gene therapies, aren't they? That's jiggling around with the body's
gene therapy. Yep.
Jiggling around with things that really aren't understand, with emergent behaviours that well, maybe people like you could anticipate, but I certainly couldn't.
Well, on that point, if we look, okay, this is a scientific paper from Pfizer scientists, and the plasmid utilized prior plasmid DNA manufacturing technology expertise from Pfizer's gene therapy program.
Right. So Pfizer used gene therapy plasmid, yeah.
Yeah, and this I learned off of a good friend and colleague, Maria Gucci. So, yeah. And it's see, it's Pfizer's own gene therapy program, right?
The recklessness here when we have all this information is just beyond description in my mind. Just outrageous corporate reckless behavior with several billion human beings. Right. Excellent, David. Anything else you wanted to bring up particularly?
I
think that's enough to cause quite enough alarm for the time being, but do tell us if there's anything else.
I think that's all. I'm very grateful for the counselors at Port Hedland,
Western Australia,
who have seen my work and are alarmed and going, this needs to be answered and looked into, and caution before more people are harmed and
transfected and harmed.
Yeah, and those counselors had courage, and I'm thankful for people like them.
It's just the experimental nature of what has been done to us is just beyond my ability to have imagined a couple of 2-3 years ago. I just can't believe how reckless this has been. It's just
right.
Yeah, and the people that were constructing these vaccines, presumably they had access to virologists of your caliber that they could have talked to if they wanted to.
I would think so.
Yeah, so would I. Great, Dave. Have you got any plans to do any more assays in the pipeline?
Yeah. Well, the whole new the vaccines are being rolled out.
Oh, the RSV, respiratory syncytial virus.
No, the SARS-2 vaccines for JN2.
Right, whatever the latest thing is, yeah.
So I'd like to check those. And two, lot of our vaccine injured people need help.
Yes.
They can't get the right tests. There's no government available spike protein test to work out levels and loads, and therefore I am developing such a test to help those who are vaccine injured.
So you're developing a spike protein assay for like a blood test.
Yes.
Wow.
Blood and urine.
If you were given a tissue sample from someone with a vaccine injury symptoms, or indeed anyone, would it be possible for you to do, or one of your colleagues to do, a genomic analysis and definitively identify the presence of foreign DNA or SV40 in those genes? Presumably, if you did a full gene sequence, that would come up.
If it's there, we can find it.
So why isn't that being done?
Well, first of all, our governments aren't funding this sort of research. I've been out of a full-time paid job for 18 months as I've worked on this, and the governments aren't funding, I'd say, rebel science.
I'd say proper scientists myself. But
Exactly. Yes. Thank you.
So how are you?
Proper science.
How are you surviving, David? If you're not,
I'm getting by barely on court cases as a expert witness and some contract work, and yeah.
you're a man who's put proper science above his own financial well-being, and a lot of people
Yes.
watching will find that a noble thing, David.
Thank you.
And I certainly appreciate what you're doing. It's impressive science. I've learned a heck of a lot of science. This is precise, detailed science. I'm no expert in it at all, but to have people of your caliber on to explain it with authority and gravitas really is helpful, and also alarming in equal measure, it must be said. And to know that you're doing this against the opposition, that proper science now can only work going uphill against official opposition is just not the way we want our democracies to be. This is outrageous.
Yeah, and so if your this and ers will want to help support this work and help things get set up and go it would be very much appreciated. I can't talk as well as the rest, except I do excellent, transparent, proper science.
Yes, absolutely. Yeah.
What is most important.
Yeah, your science is excellent, and you're able to write up the results in completely world leading reports. There's
Thank you.
no question about that. Will you send us links for those things, David, so people can get involved in this if they want to?
Yeah, I'm on X, and I'm on Substack. I will send you the links to post with this.
And they will all be in the description, so people can see those with the references as well.
Yes.
Excellent, David. Thank you so much.
You're welcome. I
know it's the middle of a busy day, but
great.
it's a lot of people are going to be pretty fascinated by this and alarmed. And I must say, I didn't realise things were as bad as you've just described. It's really helpful.
If you want, I would dive next into the replicon vaccine, which is being launched in Japan as of last week. It's a self-replicating replicating mRNA vaccine. So
I know nothing about that. So yes, if we could do that at some point, yeah, that would be good. Yeah, I've known nothing. I've heard about it, but I don't know anything about it.
If you
whatever it is, I don't like the sound of it.
If you think this is bad, that's even worse.
Right. Well, we have to learn about it. It's the nature of reality. So yeah, let's do that. Let's get this one done first. I've written out quite a long, quite a substantial pre-say of your work, which I'll use as a summary. And but for now, David, thank you so much for this talk and for your self-sacrifice in doing proper science to the benefit of humanity. We really appreciate it. Thank you. Thank you.